most of you know me already. I think parts of you already joined our morning walking tour through the city and have seen a lot of foggy British weather here. So I thought, okay, the river was visible, so that's the nice thing on that. What we have prepared in this conference is actually a program where you can interact, where you can be creative, when you can share experiences, where you can talk to each other. And actually this is the idea of the whole conference, that you actually come together and talk and just try to be innovative, to discuss things, to get new ideas, to get new insights from other people, from other disciplines, maybe from other countries. So we see we have a lot of different countries present here, and use the time to connect. And I think we will have some nice tasks today. We have a keynote just in a second from James Moore. I will introduce in a second, and we have a nice working session later on with some startups, and another keynote from Professor Duncan later on, and today in the evening the Ignite poster session, and actually the same structure more or less today and tomorrow in the we have some presentations from your submissions and come together. So just enjoy it and get connected. And now I want to introduce our first speaker. So it's Professor James Moore from Imperial College in London. And I just told him that we actually have a little bit the same background. So I also studied mechanical engineering, and he also studied that in Georgia, in the US, and was long time on the Georgia Tech Institute for mechanical engineering and bioengineering, and responsible there for some research on the vasculatories, especially aorta and coronaries. And now he is, since 2003 already, or you're in London, or it's 7 years. Yeah, 7 years in London. Okay, so and is one of the famous medical device inventors there, as I read, and also co-chair of some startups, so actually involved in also these innovation and medical device creation scene. So I'm really interested now in your talk, and I think we will talk a little bit about your experience, how to interact with medical doctors and come up with new ideas. Yeah, so thank you for being here.
Okay. Good morning, everyone. Thank you for being here, and thank you for the kind introduction, Axel. It's wonderful to be back in Magdeburg. I was here a few years ago, and I already see a big difference in the city. It's beautiful. It's amazing the amount of sort of construction going on, and you get a real energy here that wasn't present a few years ago, and it's wonderful to see. So it's great to be back here. So I'm going to tell you today about some of our research on the lymphatic system. It's a very important system, as you'll find out, and there's some interesting things that happen when diseases develop associated with the lymphatics. Now, because not many people know anything about lymphatics, I usually have to give some basic introduction of what it does. So the lymphatics run roughly in parallel to the veins. So arteries take blood away from heart, veins bring it back. But at the capillary level, which is the real business of the cardiovascular system, is delivering oxygen, nutrients to the tissues. At the capillary level, as part of doing that business, there is a net leakage of fluid out into the tissues. Now, that fluid has to be gathered up and returned to the blood system somehow. or your tissues just swell up. And we see that all the time when there's dysfunction with the lymphatic system. So the initial lymphatics are very porous. They let pretty much anything in, all of this fluid, the proteins that are in the interstitial spaces, dirt, debris, cancer cells. There are lots of things that can come into the lymphatic system at the initial level. And then the lymphatic system actively pumps that back up to the fluid return point is in your shoulders here in the subclavian veins. So most of the lymphatic system is fighting gravity when it is pumping that fluid back to your shoulder. Now, in terms of raw numbers, you can see how it compares to the blood system. So you have about 5 liters per minute coming out of your heart of blood, but the whole lymphatic system only pumps about 5 liters per day. So it's a very low flow system. It's also a low pressure system. So that's not a misprint. There are pressures in the lymphatic system. Subatmospheric pressure is down to negative 50 millimeters of mercury. Right? So a lot of the lymphatic system is actually working at subatmospheric pressure. If you poke in to the lymphatic vessel in the arm, for example, it will actually pull fluid in from the outside. So it's a fascinating system in that it pumps fluid, essentially water, through your entire body, much of it working at subatmospheric pressures. The largest lymphatic vessel is only about 2 or 3 millimeters in diameter. It runs up the middle of your thorax here. It's called the thoracic duct. And that's the thing that dumps the fluid back into your subclavian veins. So it's about an order of magnitude smaller than your largest blood vessel. So those are some basic numbers for you. Now, in terms of the role in disease, well, here's some not so fun facts. So cancers, all of the deadliest forms of cancer spread through the lymphatic system. And when they do, those cells go to another part of the body and they set up what's known as a secondary tumor. And it's 90% of all cancer deaths are due to those secondary tumors. So a breast cancer patient typically doesn't die because of the tumor in the breast, but because it's spread to the brain or the bone marrow or the lungs. Right? 90% of all cancer deaths. So if we could stop cancer from getting into and through the lymphatic system, that's a good reduction in 90% of cancer deaths. Really hugely important. Also, surgeons therefore remove lymph nodes in order to stop the cancer from spreading. And that has some problems, as you'll see later on. All of your lipids are taken up through the lymphatics in the digestive system, so that means that lymphatics play an important role in all vascular disease. It's also any autoimmune disease like rheumatoid arthritis, lupus, all of these other diseases that are involved with the immune system. That involves the lymphatic system as well because the immune system relies on the lymphatic system transport to do its basic function. It plays a role in Alzheimer's, that information is emerging now, and obesity. So right now, in this one slide, we have 8 of the top 10 causes of death in most of the world. Right? But I've been telling you so far, we don't know much about lymphatics, and it's true. And I think this tells you why. So there was a surgeon in California who was working on lymphatics, and he decided he had trouble finding any of his colleagues who knew anything about the lymphatic system. So he did a survey of medical schools all over the United States, and he said, how much time do you spend training your medical students on the lymphatic system? Right? So your engineers, T is a unit of time. I will tell you what it is in a moment. You have to guess, right? You have to come up with a guess in your head for what the unit of time T. So 53% of medical students receive this unit of time of training on the lymphatic system amongst 4 years of postgraduate education. 2 times T, 3 times T, and 1. So 53% less than unit time T. So, and that's amongst 4 years. So raise your hand if you had 30 minutes. That's incredible, isn't it? Such an important system, and they spend less than half an hour learning about it amongst 4 years of medical school. Now, when I've shown this slide to my colleagues in the UK, they all nod and say, yeah, that sounds about right. That's what they have too. And I think, unfortunately, this is true of many medical education systems around the world. We just don't learn that much about the lymphatic system. So, as I told you earlier, cancer surgeons often remove lymph nodes to stop the spread of cancer. They do this especially for breast cancer patients. So they'll take out the tumor and then they'll find the nearest lymph node that was draining that tissue, and they'll often take out that lymph node or more lymph nodes. So we have about 500 or 600 lymph nodes in the body total. You have about 30 or 40 of them underneath here, right? So if they take out only one lymph node, the chances that the patient will end up with a swollen arm are somewhere between 5 and 10%, we think. If they take out 4 lymph nodes, chances are about 50-50 that the patient will end up with a swollen arm. And if they take out all the lymph nodes, which they used to do, then it's guaranteed the patient will end up with a swollen arm. Now, this is a disease called lymphedema. It can occur naturally as a result of genetic defects. This is called primary lymphedema. Thankfully, it's fairly rare. Secondary lymphedema due to injuries or surgeries like this is actually much more common in developed countries. So we think there are something like 3 million breast cancer patients with this form of lymphedema in their arm in the USA. There's something like 10 million patients total suffering from lymphatic diseases, lymphedema of another part of the body, for example, due to another cancer surgery. So that's more than HIV, Parkinson's, multiple sclerosis, all these terrible diseases combined. You've heard of all of those diseases. Chances are you never heard of lymphedema. It's crazy. Now, in the developing world, there's a parasitic infection that can cause lymphedema, typically in the lower limbs. And we think, those numbers are hard to measure, we think there are something like 200 million, maybe 300 million people in sub-Saharan Africa and Asia that have that form of secondary lymphedema. It's affectionately known as elephantiasis, and if you Google that, you'll see some horrible images of how people have to live with swollen legs in this part of the world. So 300 million people, that's the whole population in the US with the same incurable disease. It's a massive, massive health problem. Now, as I mentioned earlier, 40 lymph nodes in the axillary region, even removing one, which is sort of the minimal that they do nowadays, can lead to this condition. And all totaled, so the rough numbers are, if you don't remember anything from the talk today, 1 in 7 women will get breast cancer. 20% of all of them will end up with a swollen arm. That's a lot of patients. And it's incurable. And it adds about $11,000 per year to their health care for the rest of their life, because there isn't a cure for this. Right, so what do we do for them? We wrap them up in compression bandages. This is uncomfortable. They have to wear them all the time. It has to be applied appropriately, and in some patients that keeps the disease from getting worse. So with compression therapy, good skin care, exercise, manual lymphatic drainage, which has to be done by someone specially trained for this purpose. Those 4 things combined are the best we can do. It's called disease management, not disease curing or disease treatment. Because it only can stop the disease from getting worse. There is no cure. So, how does the lymphatic system work? Well, it's a pumping system. Again, it's pulling tissues from pulling fluid from subatmospheric tissue beds and returning it to your shoulder. So it does this with a combination of squeezing and check valves. So that's what the check valves look like. They're bilethal check valves. They look like venous valves. And this image is from a rat mesenteric vessel that's about 100 microns in diameter, or about the diameter of the human hair. Remember, these vessels are generally very tiny in the body. Now, this is what the flow actually looks like through the lymphatic vessel. This is, again, a rat mesenteric vessel. And you will see now the importance of the lymphatic system to immune function. All of these cells inside this lymphatic vessel here are immune cells. They're either lymphocytes or dendritic cells. And they control your peripheral tissues for bad things that might hurt you. And when they pick up something bad, they go to a lymph node via this system here, and then that's how adaptive immunity happens inside the lymph node. I'll come back to that later. Now we can track the diameter of the wall of this vessel, and we can track the cells going through it, figure out their velocity, which is on the order of millimeters per minute, and we can estimate the flow rate. So the flow rate in that vessel is about 200 microliters per hour. So think about that. How much is a microliter? Piled 200 of those, it takes a whole hour for that amount of fluid to move through this lymphatic vessel. Small flow. Strangely enough, the peak wall shear stress in this vessel is about 5 dynes per square centimeter, which Axel can probably tell you is about the mean shear stress in your aorta, which is that big and has 5 liters a minute of blood going through. Some interesting biomechanics there. So this is how you get fluid from your toes up to your shoulders, is this combination of squeezing and check valves. It's essentially the way the lymphatic system works. So you can see that's what all pumps do. They take fluid from down here and they move it up here. Now that pressure difference varies in the body, obviously, and therefore the amount of flow you can generate with that pump varies as well. So mechanical engineers like Axel and myself have been studying pumps for many hundreds of years now, and this relationship between the pressure difference and the flow rate is expressed in a pump curve, and we can do similar type analysis on lymphatic vessels to see how they perform. So we can actually do, and I'll just mention the external pressure is going to be very important, so I'll bring that up later. So we can actually do this experiment. We can cannulate lymphatic vessels with pressure reservoirs and raise and lower those pressure reservoirs to see how the vessel pumps. So that's what we've done in this experiment here. These are my collaborators in the US that do these experiments. Remember, this right mesenteric vessel is 100 microns in diameter, about the size of a human hair. And my hands shake too much to do these experiments, but they can cannulate these vessels. They can put tiny tubes inside these vessels and then feed them with pressure reservoirs, raise and lower the pressure reservoirs. So what you can see here is, let me just pause this. So what you can see is the lymphatic vessel, there's a valve here, there's a valve here. The states of the valves are shown in these two curves. So the red valve is downstream, 0 is closed, 1 is open, and the blue valve is here. These are diameters from tracking the wall here and downstream, and then these are the pressure reservoirs. So initially, the two pressures are at the same level, and then what we're going to do, and you can see it happening already, we're going to raise that downstream pressure. This red curve down here shows that the downstream pressure is being increased. So we're just doing that ourselves and challenging the vessel to pump against that adverse pressure difference. You'll see the black curve here is the pressure being measured inside the vessel with this tiny needle. And as the experiment goes on, you'll see how the vessel responds to this increase in pressure challenge. So as we raise the downstream pressure, the vessel starts to contract with more force and at a higher frequency. It is sensing its mechanical environment and deciding that it needs to pump more vigorously against that increasing downstream pressure challenge. Right? Now, at about this point, we have exceeded the pumping capability of that pressure, so that downstream valve is now closed. It doesn't open anymore. The vessel is not successfully pumping anymore. So now we found the pumping limit of this vessel, which tells us a lot about it. Then we return the pressures to where they were before. Both valves go to the open position as a safety mechanism, and the vessel stops pumping. Again, it's sensing its mechanical environment. If there's no pumping challenge, it doesn't contract, except it's going to get bored and contract again there. So this is a great experiment to tell us something about the pumping dynamics of lymphatic vessels. Now what I've shown you so far is two kinds of experiments. One in the body where we can track the cells in here and measure flow, but if we try to poke in and measure pressure, we disrupt lymphatic function. So we can't measure pressure there. In this experiment, we can determine the pressures, but we can't measure the flow. If we try to put anything in here to track the fluid movement, it clogs the pipettes, the tiny tubes used to cannulate these vessels. So we can't really measure flow. We succeeded in 1 or 2 experiments. It's a lot of trouble. So we can't really do that. And in humans, you cannot measure pressure, flow, or diameter in any lymphatic vessel without disrupting. All right? So how do we learn anything about the lymphatic system? Imagine if there were no way to measure blood pressure or flow in any blood vessel. How much would we know about vascular disease? Right? So that's one of the challenges that we have for the lymphatic system. Now, one way that my group has addressed this is by doing some mathematical modeling of lymphatic pumping. So we take that information we can get from the experiments and we feed that into a lumped parameter model. This is a sort of an electrical circuit equivalent model of a lymphatic vessel. So it's got a capacitor that is active and squeezes, and then the valves are variable resistors. Right? So we input the contraction of the vessel here. That reduces the diameter, which raises the pressure, pushes flow downstream, and then this is actually the flow into the first valve upstream. Now, remember, each individual lymphatic vessel is like a little heart, right? So there's an incoming, like a mitral valve, and an outgoing, like an aortic valve. So this thing squeezes. So when it squeezes, flow goes out the downstream valve, but when it relaxes, it needs to be able to suck in fluid from upstream in order to fill during diastole and contract again during systole. So it won't have any fluid to pump unless that fluid is coming in when the vessel relaxes. Absolutely crucial to lymphatic function. People used to think that it was just contraction that did the whole thing. Actually, it's the relaxation part of the pumping cycle that we think is more important. Now, in this simulated experiment, the transmural pressure, the difference between internal and external pressure, is positive. So the internal pressure is about 5 centimeters of water. The external pressure is 2. So there's a transmural pressure difference of 3 centimeters of water there. Now, if we reduce that transmural pressure to a negative value, so now the internal pressure is less than the external pressure. Now these vessels are being compressed from the outside. We prescribe the same vessel contraction. The diameter doesn't change because it's already shrunk down. The pressure doesn't changes by a little bit. There's no flow coming in. So it cannot relax in diastole and suck in fluid from upstream. Therefore, it's got no fluid to contract during the next cycle. Right? So then the pressure volume, if you like, then becomes this. This is a dysfunctional vessel. Right? So compressing the vessel from the outside actually shuts down lymphatic pumping. Right? Now, I'll remind you, all right, so we can investigate the varying the external pressure. So here's the external pressure. So if we raise the external pressure, the amount of flow that we can generate with this pump goes down to 0. But actually, if we pull a slight vacuum on the vessel, then we can generate a lot of flow. Okay? So what happens is that on this side, the vessel is already compressed by the high external pressure, and when it contracts, it just can't generate any stroke volume. When the outside pressure is slightly negative, then that vessel can open up during its diastole, and then when it contracts, it's got a lot of fluid it can push forward. Right? But if we pull too hard on it, then it has trouble contracting. So there's actually an optimum external pressure slightly on the vacuum side at which these vessels generate the most amount of flow. Right? So compression makes the flow go to 0. Slight vacuum makes the flow optimal. Right? Now I'll remind you what we do for patients with lymphedema. We compress the limbs. Now this does help the swelling. It helps prevent the swelling from getting any worse in these patients. But it's not a cure, right? So what we think would be maybe a better thing to do, and I wouldn't do this all day, but for part of the day, maybe 20 or 30 minutes per day, apply a slight cycling vacuum on the outside of the arm. So here is the very simple device. Here's the patient's arm here. There is a seal near the shoulder level. And by turning these pedals here like a bicycle, we can create a cycling vacuum inside this tube and hopefully improve the flow of lymph out of these patients' arms. Now, we've tried this out on some healthy students like yourselves. I tried it out on me. My arm is very skinny already, so it was hard to pick up any change in volume, and most students are very healthy and their arms are not huge. But so we're getting to the point where we're getting ready to try this out on patients now. We're very much looking forward to that. Now you might ask, why do we have this pedal thing here? Why not have just an electric motor or a pump on there? Remember, there are something like 200 to 300 million people worldwide with lymphedema. We want this technology to be available to them as well. So that's the reason it looks so simple. So that's one of the devices that we have under development for patients with lymphedema. Remember, there is no cure. These patients are absolutely desperate. They'll try almost anything to get the swelling down in their arm. And we think we'll have them lined up for kilometers around the building whenever we decide to test this thing out. So now others have tried applying slight vacuum on the outside of the edematous tissue, but they don't really know how to adjust the vacuum. They don't know what pressure to apply, what frequency, and so forth. It's with our computational modeling now that we can test different amplitudes and frequencies of cycling pressure on the outside of the arm, and we can figure out what's the optimal regime to apply to these vessels. So we're well on our way to getting that out there to where it can help patients. The other device that we have under development to help patients with lymphedema is an implant that would go in at the time of surgery in the place of the original lymph node that's being removed. So this implant would include a growth factor that would encourage the vessels to grow to it, but also it incorporates in its design some intelligent fluid mechanics. So here is a basic picture of a lymph node. We'll go into more detail about lymph nodes shortly, but as vessels approach a lymph node, they split into multiple parallel vessels. Now remember, each individual vessel that's doing its pumping can only generate a few centimeters of water pressure. Right? And then this lymph node is this massive sponge. It's a big resistor in the way of the flow coming out of those vessels. One way that pumps can get around big resistors is by constructing multiple parallel pathways. So if you're an electrical engineer, this is like putting resistors in parallel with each other. The total resistance presented to the system is inversely proportional to the number of resistors. So it's a way of reducing the resistance to the flow coming in from these tiny little pumping vessels. So our device has multiple parallel pathways. These are sized for a rat lymph node. They're tiny. Lymph nodes in humans are much bigger than this. So we have 3 parallel pathways in this vessel here for good fluid mechanics and the growth factor to encourage the vessels to regrow. So we're on our way to developing that technology. You can see what it looks like here where they converge, the pathways converge into one. We are already at version 2.0. We're getting ready to try that out in rats and eventually in larger animals and then in humans. And then once we hook into the superhighway of the immune system, we think we can use these implants to restore or modulate immune function, which has implications on many, many, many diseases. So our vision really for this implant is that it gets used in every lymph node harvesting surgical procedure. Most of these associated with cancer. Now there are 100,000 procedures per year in the US, which gives us a big enough market, we think, to justify this as a reasonable sized business. But I'll remind you, there's no way to tell which of these patients is going to get lymphedema. So really our target market is not the 20% of patients that get it, it's 100% of patients that get these surgical procedures because we never know who's going to get lymphedema. The implant itself is made from polyurethane, so it would be as safe as leaving sutures in the body. So that's where we're headed with that device. Now I'd like to switch a little bit from talking about lymphedema and talk about more the immune side of lymphatic function, in particular vaccines. Now hopefully we've all had vaccines. If you haven't, leave now and go get a vaccine. It's becoming a serious problem. So vaccines typically include some version of the antigen that might kill you, right? So it's typically some dumbed down version that won't kill you, but it's a way of teaching your immune system about the bad antigen that could be dangerous someday, and once your immune system learns about it, then this process called adaptive immunity happens, and then you're prepared for it whenever you encounter it again. So that's the antigen. Now, often vaccines also include a substance called an adjuvant, which is meant to kick up your immune response and increase the recognition of the antigen. So these adjuvants have been around for a long time. First one was discovered by accident in 1920. Since then, aluminum salts, or alum, have been used in over 10 billion vaccines around the world. Almost all of us have had a vaccine that includes alum, right? Remember, nobody knows how it works, right? It was discovered by accident. It wasn't a very rational decision to start including these in vaccines, but studies have been shown that have been done that show that if you use an adjuvant with the vaccine, typically it makes the vaccine response better, right? Now, you think about the influenza vaccine. Well, at best, influenza vaccine is about 60% effective. So if we all got the flu vaccine this year, for 40% of us, it does nothing. Some years, it's only 20% effective. So for 80% of us, it would do nothing. Right? So can we increase those effectiveness rates of vaccines just by understanding adjuvants better and doing something about it? So that's kind of where we're headed with this work. So here's the process. Generally speaking, is you have the antigen and the adjuvant in the vaccine here in a syringe. They typically will inject that intramuscularly. There are not many lymphatics in your muscles. Most of the lymphatics are further out in the epidermal space or the subcutaneous space, but that's where we inject vaccines typically. Those lymphatics pull in the antigen information. They go through the pumping lymphatics that I've shown you earlier, and then goes into a lymph node where that's where actually adaptive immunity happens, is inside the lymph node. Right? So you need to get that antigen information into the lymph node. That should be the goal of the vaccine. Now, most of the people that have been studying vaccines and adjuvants over the past 100 years or so are biologists, and I'll tell you something about the way a biologist views this process. And if you're a biologist in the room, I apologize if this makes you sound bad. It's not intended that way. It's just the truth about what we know so far. So the way adjuvants make vaccines better, there's a theory called the depot theory, which is that at the point of injection, adjuvants increase the recognition of your local immune cells in the injection point to the antigen, and then they take that information out, and somehow that teaches the immune system better. So that was actually the person who developed aluminum salts to begin with, came up with the depot theory himself. He had no proof for it. This is still the dog of the day on how adjuvants work with vaccines. Now, a few years ago, there was an experiment done where they injected a vaccine in the ear of a mouse, and then 30 seconds later cut off the mouse's ear. Changes nothing, right? If there were a depot effect, you would need to leave that ear intact, right, for that vaccine. You cut the ear off, you still get an immune response. So within 30 seconds, something has happened that takes that vaccine information into a lymph node, and you would develop adaptive immunity just fine. So the depot theory has been largely disproven, but you still hear people talk about it. What else have we got? Well, biologists have also imaged dendritic cells, or antigen-presenting cells, that live under your skin, and when they take up antigen, they start to crawl into a lymphatic vessel, those initial porous lymphatics, and they crawl along the wall of those initial lymphatics at a velocity of about 5 nanometers per hour, if I remember my units correctly, okay? And so the biologist says, well, that's the way the information gets to the lymph node, is the dendritic cell crawls along the wall of the lymphatic vessel until it gets to a lymph node. At that speed of 5 nanometers per minute, it would take 4 years for that cell to go 1 centimeter. This is not what happens, right? This is not how the process happens. But this gives you some idea of how little we know about how these things perform. This is the dogma, Finally, inside the lymph node, actually, there's been a lot of very, very nice work on what happens to vaccines and adjuvants when they come inside the lymph node. There's size-selective filtering, so small antigens will go directly into the meaty part of the lymph node. Larger antigens have to be ushered in by cells. It's a fascinating little system. But no one's ever looked at the role of transport in any of these processes, much less lymph nodes. So we started a few years ago trying to understand lymph nodes and the effects of adjuvants on flow through the lymph node. So that's what I'm going to tell you about today. And so here's the lymph node structure, a typical cartoon that you would see in an anatomy textbook. So there are typically multiple incoming lymphatic vessels. They come into what's known as the subcapsular sinus, which is an open region. So it's like a parallel plate with fluid flowing in the middle of it. And so the fluid comes in from these incoming vessels, gets distributed around the outside of the vessel in the subcapsular sinus, and then some of that fluid will flow into the middle, which is more like a porous medium. So in red, there's a blood vessel running through the middle of the lymph node. And that porous medium then is sort of like a sponge, and some of the fluid will percolate in, but that porous medium contains all of your immune cells. All right, so that's where adaptive immunity happens. That's where you need that antigen information to be delivered. Right, so then how does flow look through the lymph node? Well, it can come in from it comes in from the afferent vessel. It can either go directly around the outside from the subcapsular sinus to the medullary sinus, that's shown there in the light blue. B-cell follicles, B cells are one type of immune cell. The T-cell cortex is in the middle. T cells are another type of immune cell. And those cells need to communicate with each other in order for adaptive immunity to happen. There are also blood vessels inside lymph nodes. I'll come back to that later. So you can see literally the flow chart here is the fluid comes in, some of it can go around the outside, some of it can go into the middle, and nobody had ever looked at the distribution of these flow patterns before we did it a few years ago. And so what happens is, and we've done our best to, of course, base our model on all the experimental data we can get our hands on, and some that we generated ourselves. So it seems that under baseline conditions, when there's no immune response going on, about 90% of the flow goes around the outside of the vessel. It's following the path of least resistance. Remember that subcapsular sinus is like an open channel for flow. And so that's going to be the path of least resistance. The medullary sinus is also fairly open, but then you've got that big tight sponge in the middle. So the flow is preferentially going to follow that path of least resistance around the outside. Now, the flow that does come into the middle sort of gets absorbed by the blood vessels on the inside here. But that's only about 10% of the flow, again, under baseline conditions. So, under baseline conditions, you only need to smell about 10% of what's coming in. Just make sure there's nothing wrong, and then let the fluid go on through to the next lymphatic vessel. So now we wanted to see what the effect of adjuvants are on those flow distributions. So we did an experiment with some mice. So we injected an adjuvant in the foot pad of the mouse, and then we took out the lymph node either behind the knee or in the groin. We could do it either way. And the mouse lymph nodes are about 1 millimeter in diameter. Your lymph nodes are about 2 centimeters in diameter. So mouse lymph nodes are tiny in comparison. And then we cannulate them. Again, these vessels are smaller than a human hair. And the flow rates through these vessels, remember, are hundreds of microliters per hour. Pressure differences are a few centimeters of water. This is an absolutely exquisite experiment to try to do. You have to measure tiny pressure differences, tiny flow rates after cannulating tiny vessels. Right? So, so that's what we do. We measure the flow resistance, so we vary the pressure across the lymph node, and we measure the flow rate that it allows through it after we've injected these different vaccine adjuvants. So this is what happens to flow resistance across the lymph node. Under baseline conditions, your lymph nodes are fairly low resistance to the incoming flow. So all of the healthy lymph node resistance measurements were down in that neighborhood. Within 24 hours of injection of the vaccine, the resistance of the lymph node has gone up by an order of magnitude on average. You can see over here. Now that's before the lymph node has started to swell, right? So you know your lymph nodes swell when you get sick, right? So the swelling happens generally after this point. So the lymph node has recognized that there's a problem and is shutting down the flow. It's increasing the flow resistance, shutting down the flow. So then we went back to our computational model to try and answer what part of the lymph node is responsible for this resistance increase. So we looked at a few different things. We can, it's a great thing about computational modeling is you can do all kinds of variations in the geometry and the parameters. So we varied the porosity of that big, meaty, central, spongy part of the lymph node. But remember, most of the fluid is going around the path of least resistance, not through the middle. So if you change the resistance of the big spongy part in the middle, it does nothing to the flow. Right? So what about that subcapsular sinus? Some people think that maybe the subcapsular sinus is getting bigger or smaller as the flow comes in. So we vary the height of that open channel region around the outside, and that's how much effect it had on flow resistance. So that's not it either. The last thing we did was we varied the porosity of the medullary sinus. So I'll remind you, the medullary sinus is down here. It has a direct connection to the subcapsular sinus and a connection to the T-cell cortex as well. So this is sort of the outflow region of the lymph node. And when we varied the porosity of the medullary sinus, at that point, we could recreate the experimental results showing an order of magnitude increase in flow resistance. So there must be some cell type in the medullary sinus, or I think perhaps at the connection between the subcapsular sinus and the medullary sinus that creates, that shuts down that low resistance pathway, right? That's what increases, that's the only part of the node that we found that can increase the flow resistance overall, right? So, so what does that do then to the flow pattern? Well, it increases the flow into that meaty central region where all your immune cells are by threefold. All right, now you've got something that makes sense, right? So only 10% of the fluid is being exposed to your immune cells under baseline conditions. But when you've got a signal that there's some antigen coming in, that path of least resistance shuts down. And 3 times more flow is now being shunted into the middle part of the lymph node, where your body can learn about the bad things coming in. Right? And again, this is, I think, a nice example of how we can combine experiments with modeling to reveal more insight than we would have gotten from either one in isolation. Right, so again, baseline conditions, 90% of the flow around the outside, 10% of the flow into the middle. And then what we want to do is then look at what happens to that 10% of the flow that comes into the middle part. Now, I told you it gets absorbed by the blood vessels there, right? And I just kind of hand waved it away. It's actually a really important process. We think Starling's equation is a reasonable approximation to this process. So I'll walk you through this. L is the hydraulic conductivity of the blood vessel wall. So that cylindrical surface around the outside of the blood vessel. Area is the blood vessel surface, so that's 2 pi times the radius times the length. It's like the outside of a beer can, right? And then that limits the amount of flow, those 2 factors together. The driving force for the flow is the difference in lymphatic pressure and blood pressure, but also oncotic pressure. So this is the protein concentration difference, like osmotic pressure. Oncotic pressure is just osmotic pressure for protein. So that's what determines the direction of flow. And what we think, based on a lot of experiments done in the 80s by Guyton, famous physiologist, and what we've simulated as well, is that there's fluid moving out on the arterial side and back in on the venous side due to the difference in the blood pressure. Now, We know what blood pressure is. We know what lymph pressure is from the previous computational simulations I showed you. The lymphatic pressures are easy to get at. But the thing that we don't know is the distribution of the blood vessel surface area or the blood vessel pressure as it varies going from the arteries into the veins. And there are no images that we could find of blood vessels inside lymph nodes. So we did that. We did some micro CT, and this is, remember, a mouse lymph node is about 1 millimeter in diameter. So this is a 1 millimeter lymph node. And you can see the blood vessel distribution inside there. This is coloring coded by diameter. So you see one of the big red vessels in the artery, the other big red vessel is a vein. Most of the vessels that are in there are capillaries, and they're dark blue, right? So it turns out most of the capillaries, which presents most of the surface area, is distributed peripherally around sort of the outside, the outer part of the T-cell cortex. The middle of the T-cell cortex actually has relatively few blood vessels going in through it. And we've redone the computational model and shown how that affects the lymph flow patterns inside the lymph nodes. Now, remember, your lymph nodes swell when you get sick. The blood vessels inside your lymph node also undergo an incredible process of angiogenesis. So this lymph node blood vessel structure will completely change within days after vaccination or some kind of infection. And it also changes when cancer cells come into the lymph node. And we're working like crazy to try and image those blood vessels. This image, a single image here, took a few days of work. Not just for the micro CT imaging, but you get terabytes of data from the micro CT scanner that we use, and it takes a while to get these images. So, and we calculate the blood flow through them as well. So there's a lot we can do with the information once we have it, but it's a lot of work to get the information. So, I'll take you back to this picture. Remember, we want the antigen information to get from the tube, from the syringe, into the lymphatic vessels here, down the pumping lymphatic vessels that I showed you earlier, and then into the lymph node. So much of what I told you in the last 10 or 15 minutes has been associated with what happens inside the lymph node. Fascinating little structures, and we can't wait to learn more about them and what those adjuvants do to that delivery process. Now, what I'd like to close with is to show you some of our brand new results. This is unpublished stuff. Of what the effects of adjuvants have, and we've tested all 3 types of adjuvants. So there's aluminum salts there. This is oil and water emulsion, like plant oils. Literally, the adjuvant design is, hey, let's poke some of this in and see if it works. That's the entire adjuvant design process. And then these are toll-like receptor ligands here. So the 3 types of adjuvants. And we've actually studied the effect that they have on the pumping of those lymphatic vessels. Now remember, if an antigen-presenting cell has to crawl all the way to the lymph node, it's going to take years, right? So the way it has to get there is getting pumped along these lymphatic vessels that I showed you earlier with the pumping and the squeezing in the valves, right? This is what adjuvants do. Every adjuvant we've tested so far shuts down lymphatic pumping. It interrupts the muscular contraction of the lymphatic vessel wall. So that's what happens to the diastolic diameter, so these vessels just swell up. And that's what happens to the contraction frequency. They essentially stop contracting with adjuvants. Doesn't that seem like the opposite of what you would want to happen? Who knows? So really the way to think about this is, what does the lymph node need? Does it need a bolus and then a steady supply of the antigen? Or does it need a gradually increasing supply of antigen? What does it need? Nobody really knows, but the right way to think about this is start with the lymph node. Think about what needs to be delivered in the lymph node as a time course to optimize adaptive immunity, and then work your way backwards. And then think about what needs to happen to the pumping to deliver that. And then where in the body we should be injecting that. Probably not in the muscle, which is what we do today. Right? So this whole idea of adjuvant-induced lymphatic delivery of antigen information is something that I'm not aware of anybody else in the world trying to work on this, but I think it's a really important problem for vaccines. And I think there's a lot of room for improvement in vaccine success rates if we can understand this process better. So I'll sort of close up telling you about, we do this and more about the lymphatic system in my lab. You can see the laundry list of projects that we have going on. Now we do some experiments, we do some modeling, and those overlap to a certain degree, and you see some device development as well. So we're really trying to understand the basics of the system because remember, nobody knows anything about lymphatics. And in terms of people, the bioengineers working on the lymphatic system, you got me and a couple other people that are more or less senior type people and a couple of our former students. And that's it. I mean, I'm talking single-digit number of people worldwide, bioengineers working on the lymphatic system. So maybe we'll include some of you someday. We are trying to develop these devices. As Axel mentioned, I'm part of a medical device entrepreneurship education program at Imperial College. And so we have the students write the business plans around these devices. It's a lot of fun. So with that, so lymphatics, as you remember from my title, I call it the sewer system. So it is something like the sewer system. So whatever goes into the street gets taken up by the sewers, and then you have to clean it before you drink the water again, right? That's more or less what the lymphatic system does for your body. And remember, the initial lymphatic's very porous, like the sewers in the street. So anything comes in, including cancer cells, and a lot of bad things that can hurt you. And the lymph node is like the sewage processing facility. So there you go. Lymphedema, serious health problem, and we're doing what we can to improve it, not only with applying this cycling vacuum on the outside, but also the lymph node implant. And then the recent work that we've done on vaccine design is hugely exciting for us. So with that, I'll close up with acknowledgements of funding sources and also the people that really did the work. What you've seen today is largely the work of a couple of PhD students, Samira Jamali and Mohammed Jafarnejad, who are now at Johns Hopkins, and they really did a lot of what you've seen today, but all of these people have been incredibly valuable to these projects, and it's not something I could possibly have done on my own. So thank you for your attention, and it's a great honor to be here.
So, thank you, James. We have some time for some questions, so whoever will come up with some questions. Oh, yeah. I think you can just speak loud.
First of all, I'm very thankful to you, sir, for that, for all the information. My question is that my question is divided into two parts. Suppose if a patient is suffering from some kind of cancer, the cell get migrated to a different part of the body, right? And doctor biggest challenge is to see that where is that particular cell is there, and So and the second thing is that if a person is healthy and he's suffering from this kind of problem like edema, lymphoedema, so it is possible that we can design some kind of implant which can help us instead about the mathematical modeling, we can use white box and gray box modeling to get input and output, and we can devise some kind of feedback, so that we can get the output, and we can from that output we can create a loop to our input, something like that.
Yeah, you're right, and in some sense we're trying to put some information into some of that black box or gray box model, and so just in terms of the fluid pumping, it's actually a very complex process. But then when you include all of immunology and oncology in that process, it's massively complex. There are multiple cell types that are important for the immunological aspects of what you're talking about. So it's, yes, you're right. That's sort of the long-term goal of everyone working in this field, is to understand this, understand the lymphatics better. Also, I'll point out, remember the slide at the beginning about how medical doctors only get half an hour training on the lymphatic system? All of you now have more than that. So hopefully you can help us do this.
And second thing is that every individual human being is different. Their immunity is totally different. So how you can apply this vaccine to everyone? Because the atmosphere is different. Everyone living in different atmosphere, and their immune system is quite different. We can't generate. We can't generalize this vaccine in one particular way that this is going to work, right?
Yeah.
So, because it's a natural response to the body and it's totally different. So how you can simulate the working mechanism of our immune response with this vaccine?
Yeah, I'm sure you're right. I'm sure there's a lot of individual variability in immune response, and also in lymphatic transport. And some of that variety amongst individuals has been measured in terms of lymphatic pumping. If we can understand anything about the process, even in an average human, that's a good starting point, and that's kind of what we're working towards right now. As we gather that information, then we can start looking at individual variations and what we can do for say, people with compromised immune systems to improve their adaptive immunity. But we can't even understand one person.
Sure. Can I just ask a short question? So, as I understood right, they take out the lymph nodes and actually to stop the transport of the tumor cells or spread of the tumor cells to other organs. So that means actually create a fibers at the end. Somehow.
Yeah.
So now your system starts to pump again. Did you not start to pump the tumor cells again? Or is it?
Well, so presumably they've taken out the tumor as part of the surgery, but yeah, if there are tumor cells left behind.
But cancer is a systemic disease. I think it's not so local. So if you have it in the whole lymphatic system, actually you have to clean it before.
Yeah, well, so we all have mutant cells. Every person in this room has mutant cells. When they get pumped into a lymph node, your immune system responds and destroys them. So your immune system is really trained to deal with cancer. It's when that system is overwhelmed that bad things happen. So you're correct. They take out lymph nodes to stop the spread, but they also to diagnose the patient. So if you hear cancer in stages, stage 1, stage 2, 3, and 4, those stages are based on whether or not the lymph nodes are positive for cancer cells. So they use it for diagnosis.
Thank you very much for your presentation. I'm a mechanical engineer and I work in a mechanical engineering job. Actually, I had more than half an hour
Good.
training, but that's because I'm going to get with surgeons that treat the small lymph nodes connected to small arteries to try to solve the problem with stem now.
Yeah.
Do you think is that effective? Because you only take one small vessel or two, and then you connect them to an artery, and that solves the problems. And doubting a little bit, but he does have the procedure for a long time.
Yeah, so the procedure you're talking about is called autologous lymph node transfer. Mm-hmm. So basically what they'll do is they'll take a lymph node and its surrounding tissue from the groin or some other spot.
Or is getting that from the arm? You injected some material to see them with fluids. And then he finds them, and he shoots this with a 1/10 millimeter vessel, and that's really amazing to see on the microscope.
Yeah. Okay. All right. So I misunderstood. So that's called Venous Anastomosis. So they look for a lymphatic vessel, they connect it directly to a vein rather than the flow pathway it was using before. So that kind of surgery and autologous lymph node transfer. So they take a lymph node from the groin and its surrounding tissue and they move it up here. When they do the lymph node transfer surgery, it's about 10 hours under general anesthesia because they have to reconnect the blood vessels, which are typically about 1 millimeter in diameter. So 10 hours under general anesthesia can kill you. It's terrible. Yeah. So lymphovenous anastomosis takes a few hours. it's not an easy process. There are only a few people around the world that can do it, and it's not generally going to be paid for by any insurance, or most healthcare systems won't cover it. So the success rates are mixed, depending on the expertise of the person doing it. It's also a bit of a gamble because the lymphatic vessels in your arm are typically operating around atmospheric pressure when they're doing their pumping. Your venous pressure is about 10 or 15 millimeters of mercury. So you're trying to push fluid uphill with a tiny vessel. So I think that, you know, I wish those procedures would work 100% of the time. I would go work on something else. I have plenty of problems I can solve. And if someone came up with a cure for lymphedema, I would be the happiest person in the world. But the success rates for these things are, I don't think there's enough data yet to tell us what the success rate is, but I think it's going to be highly dependent on the surgeon doing it and which vessel they choose because of those pressure differences. So there's a surgeon.
To shoot for them, so it's already on that side, so they can do that.
Yeah, so you can image superficial lymphatic vessels with fluorescence, which is how they find the ones they use to connect. So there's a surgeon in Belgium that actually tries to measure the pressures in the vessels before he connects them. And the pressure difference is too adverse, he just moves to a different part of the arm and tries to find another vessel. But he's the only one in the world that does that as far as I know. So yeah, there are some new surgical techniques that are emerging, and I hope they work. Another thing to keep in mind is that the immune system in these patients is severely compromised because you can't move immune cells around without lymphatic flow. So doing surgery on someone with a compromised immune system in the arm is dangerous as well.
This was very awesome, my percent I attended.
Yeah, no, I'm glad you know about that. That's good.
I have also.
Okay.
Considering the functionality of the lymph node, and when we remove this lymph node and we use the implant, is it a possibility that the efficiency of the vaccine will be compromised after you insert the implant?
I suppose, yes. I mean, we are not, in the early form of the implant, we're not restoring immune function, we're just trying to keep fluid moving. If that patient were to need a vaccine later on, they could have it in another part of the body, not in the part that's been swollen. And you wouldn't want to stick a needle in their arm, in that arm anyway, again, because of the compromised immune system. So that would be a bad choice. Even without the implant, that would be a bad choice for a vaccine location. So you go to the other arm or the leg or another piece of tissue. But yeah, it's a good question.
Any questions? Okay, so let's thank the speaker again and present for you, and thank you for coming here. And it's a special wine on our university. Yeah. So from a local vineyard here, and a small women's from our university.
All right, so we would have lunch served in about 2-3 minutes. And I would request everyone to please check the notice boards after 1 p.m. for details on the working sessions. So as you know, we have 3 working sessions in parallel, and you get to choose to which one you want to go. But if you decide to go to one, you have to stay in that one for the entire duration. Okay, thank you all, and see you on the way.